here. So this is slide 6. We're going to pick up there to discuss special properties of water. And the special properties of water, there isn't a slide, a PowerPoint slide, but on your study guide, the hard copy of the study guide that I'll be giving you in lecture, with question number 5 on the study guide, it says to describe the special properties of water. So let me see if I can just hit a few of them here. And again, you'll be getting a hard copy of this. So the special unusual properties of water, what we sometimes call the emergent properties of water, it's excellent at moderating temperature because the water molecules, when they form hydrogen bonds with a lot of other water molecules, they can absorb a lot of heat energy before those bonds are broken. And if we describe temperature as the speed or the kinetic energy of molecules, this means that water can absorb lots of heat before the hydrogen bonds are broken and the water molecules start moving faster, which we would record as an increase in temperature. Water is important in evaporative cooling. Water exhibits cohesion. That's when like molecules are attracted to one another, and this would be through hydrogen bonds. We'll see that water demonstrates adhesion. This is when water molecule is attracted to unlike molecules. Water is considered the solvent of life, and we'll be describing its capacity to, for example, be involved in hydrolysis. And we'll also be discussing dehydration, synthesis or condensation when the elements of water are removed between two monomers to form a dimer. Okay, so and again, these unusual properties of water, we can describe those as a consequence of water structure. We want to remember it's bent. It's a polar molecule itself, and it can form up to 4 hydrogen bonds with other water molecules or other substances. And again, we want to remember, we said hydrophilic would be molecules that are rich in polar covalent bonds or which carry full charges. Those molecules water would be attracted to. And organic molecules that are rich in nonpolar covalent bonds, water is not attracted to those substances, and thus they're called hydrophobic. Now the next part of our unit here, we're going to do a short little introduction to organic molecules and organic chemistry. Organic molecules are those which contain carbon, but by convention we exclude carbon dioxide. We consider carbon dioxide inorganic, and all other carbon molecules is organic. I think it was thought at one time only living organisms could make organic molecules, but now we know they can be synthesized in the lab. With organic molecules, there is an incredible diversity, and so we just want to explore why that is. Well, one reason is that if we think of carbon as our building block, carbon can form 4 really strong covalent bonds. Carbon can bind to other carbons, making what we sometimes call carbon backbones or skeletons, and there's almost an endless variety of carbon skeletons that we can build. And we'll be seeing some examples here. And then we'll see, upon this rich diversity of carbon skeletons, we can add what we will call functional groups. And we'll be looking at some functional groups here just in a moment. So again, we just want to state that carbon can form 4 really strong covalent bonds, and thus these carbon skeletons will form the backbone for an incredible variety of organic molecules. And here we can see 2 carbon molecules with a covalent bond, and then to make up the total of 4 covalent bonds for each carbon, then we have 3 hydrogens covalently bonded to the carbons. And remember that the carbon-carbon covalent bond, that's going to be non-polar covalent bond, and the carbon-hydrogen is going to be a nonpolar covalent bond. So a simple molecule that just consists of carbon and hydrogen, we would say it's hydrophobic. Water's not attracted. These are just some examples of some of the examples of the carbon skeletons. So for example, we could start out by just covalently linking 2 carbons together. We could add a 3rd one and make a new carbon backbone. We could add a 4th carbon, make a brand new carbon backbone for a new organic molecule. We can introduce double bonds. We could move the double bond And with each of those changes, we have a brand new organic molecule. We can have branch chains, and I think when we start looking at the carbohydrates, we'll show you some branch carbon chains. We can move the branch point, we can change the number of carbons in the branch, and then also we can make these really cool ring structures with our carbons. So the convention here is when we show these ring structures, and at the angles, if there's not an abbreviation for a chemical element, we presume that we have carbon. Let's see if I can get my mouse. Oops, sorry. Sorry. Having technical difficulties here. Okay, let's see if we can go backwards. We do not want to go backwards. Okay, sorry, folks. Let me do an escape. Sorry for the technical difficulty here. Okay, let's see if we can go back here. Okay, sorry. My cat's also trying to walk all over my keyboard, so it's making it more challenging. Okay, no more excuses. Okay, so in our ring structures of our carbons, mouse challenges. Okay, we'll just do it simple here. Okay, in the very bottom where you see these ring structures, these are the ring structures of glucose, I believe. At the angles, if there isn't a abbreviation for a chemical element, like at this corner, this angle, you'll see there's an oxygen. Well, if there isn't an abbreviation for a chemical element, the convention is we know that carbons are present. So in this ring structure, there would be 1, 2, 3, 4, 5 carbons in the ring structure, and then there's a sixth one that's attached. Okay. And again, we can change the ring structures, we can change the number of carbons present, we can add double bonds, move the double bonds around, we can add branch points. So the point being of this slide is that using carbon as our building block, we have a limitless variety of structures that we can make. And again, we're going to see that having a great variety of carbon backbones and making it possible to have a great variety of organic molecules with lots of different functions. So if we just look at some specific examples of organic molecules, the simplest ones would be hydrocarbons, and the name tells us that these are organic molecules made up of only carbon and hydrogen. So do remember that in hydrocarbons, the carbon-carbon double bond is nonpolar, the carbon-hydrogen bond is nonpolar, and thus hydrocarbons would be hydrophobic. Water is not attracted to them. And these hydrocarbons within cells are relatively non-reactive. So the example we have here on this slide is methane. So we'll ask the question, if hydrocarbons are relatively non-reactive, what can be added to make the organic molecules more reactive or interactive? We need to have our organic molecules interacting with one another. So the answer is we're going to add functional groups, which are chemical groups that are attached to the carbon skeleton. And it's these functional groups that we could say give the organic molecule its chemical personality, and its interactions between the functional group of organic molecules, which will result in chemical reactions between different organic molecules. So I know at first this looks really daunting, but believe it or not, you guys, it's not that hard. I'll just go through these functional groups. So on the left-hand column, you can see we have the name of the functional group. And then in the second column, the big R stands for the rest of the molecule. That would be the carbon skeleton. And then you can see a covalent link to the functional group itself. So we'll just start with the simplest one. So the simplest one, the name is a hydroxyl. And the structure of the hydroxyl is an oxygen covalently linked to a hydrogen. We know that oxygen-hydrogen bond is going to be a polar covalent bond. So if we add hydroxyl groups to a carbon skeleton, we're going to make them more hydrophilic. We'll see hydroxyl groups everywhere. We're going to see them in our amino acids, our proteins, our nucleotides, and our sugars. The second one down is the carboxyl group. Okay. And this is a little bit more complicated. We'll see in the second column there's a carbon with a a double bond to an oxygen, and again that will be a polar covalent bond. And then furthermore, attached to that same carbon, there will be a hydroxyl group. So carboxyl groups are very polar, rich in polar covalent bonds. They make the carbon skeleton really hydrophilic. And furthermore, we'll see, and we'll demonstrate this on the board in lecture, that these two oxygens attached to the carbon are so electronegative, they're going to exert such an attractive force on the electron pair shared here between the oxygen and the hydrogen, that the hydrogen electron is going to get stripped off and transferred to this residue of the carboxyl group, and this hydrogen is going to be released as a hydrogen ion or as a proton. And anything that causes the increase in hydrogen ion concentration in solutions is called an acid. So these carboxyl groups are going to act as acids. The third one down and fourth row down, folks, what I wish they would have done is just given you this generic carbon double bonded to an oxygen. This is called a carbonyl group, and again, it's a polar covalent bond. If it's added to carbon backbone, it makes the organic molecule hydrophilic. And we have two descriptions. If we go into the second column for what's called the ketone group, a ketone is when we have, let's say, at least 3 carbons, 1, 2, 3, and the carbonyl group is in the middle. It'd be the middle carbon. So a ketone is when we have a carbonyl group that's not on the ends of a carbon chain, it's in the interior. In contrast, if we have if we have a carbonyl group attached to the terminal, the last carbon on a chain, then the organic molecule is called an aldehyde. Okay, so for our lecture exam, just know that a carbonyl is a carbon double bonded to an oxygen, that it's polar, and it will increase the hydrophilicity of an organic molecule. The next group here, this is a really cool one. This is called the amino group. So it's a nitrogen covalently linked to 2 hydrogens. These are polar covalent bonds, and thus they'll make an organic molecule more hydrophilic. But the other thing that's really cool about amino groups is, you don't see it here, but there's a pair of non-bonding electrons, and they have negative charges. And so if we have hydrogen ions, protons in solution, the proton or hydrogen ion can bind to that pair of non-bonding electrons, and thus we're going to end up with an ionized amino group. So these amino groups then, since they can bind free hydrogen ions, they act as bases. This last one here, not last one, I shouldn't say last one, this sulfhydryl group here, it's a sulfur covalently linked to a hydrogen, and I think we looked up the differences in electronegativity, and I think officially this would be considered a non-polar covalent bond. I wouldn't ask this on the lecture exam if I'm asking you about functional groups. I just wanted you to see the sulfhydryl group, because we're going to see it will play a really important role in stabilizing tertiary and quaternary structure amongst proteins. So we'll be talking about this later. And then this last one, this is a cool one. Now, in this table, they've called this functional group phosphate, which officially is not correct. Phosphate means that it's ionized. So what they're actually showing us here is the unionized phosphate group. One of the chem profs said I could maybe call it a phosphoric acid residue, but we see there's all these oxygens, all these incredibly electronegative oxygens here. So we have a central phosphorus double bonded to an oxygen. Here's another oxygen. We have the phosphorus covalently linked to hydroxyl group, and here the phosphorus is again covalently linked to another hydroxyl group. So these unionized phosphate groups, they're really, really hydrophilic. Water adores them, and because of all these electronegative oxygens, we're going to strip the shared electrons away from this hydrogen, releasing one hydrogen ion, and then in a second ionization we can strip the shared electron away from that hydrogen, releasing a second hydrogen ion or proton. So these unionized phosphate groups, if we put them in a water-rich environment, they are going to release 2 hydrogen ions. So that will really increase hydrogen ion concentration, Thus, the unionized phosphate groups are very, very acidic. And we're going to see these phosphate groups, we'll see them in phospholipids, we're going to see them in nucleotides and nucleic acids, and also we'll be seeing them in our energy currency of the cell, good old ATP. So this is a classic example of perhaps a lecture exam 1 question. So this is the generic formula for an unionized amino acid. So we'll just go over the parts really quickly. So with your generic amino acid, you're going to have a central carbon here, and then you'll have 4 different groups or 4 different elements attached. So we have a hydrogen attached here. Okay, and this is where the lecture exam questions would come in. If I'd circled this functional group, can you give me its name? So what's the name of this functional group? And you can check back on the table from the previous slide. This is a carboxyl group. And if I ask you about the nature of these covalent bonds, you know they're all polar covalent bonds. So carboxyl groups are going to be, and so quiz question, would a carboxyl group be hydrophilic or hydrophobic? We know it's going to be hydrophilic, right? All those polar covalent bonds. And if I ask, could the carboxyl group act as an acid or a base or neither? You want to remember that we can get a reversible ionization. The shared electron with a hydrogen gets ripped off. This guy steals it. And then that means we're going to release a hydrogen ion, a proton. The ionized carboxyl group would carry a negative charge. The released hydrogen ion would carry a positive charge. And we know anything that increases hydrogen ion concentration in solution is called an acid. So the carboxyl group will act as an acid. We'll go over here to this other functional group. So can you provide the name of this group? This is the amino group. It's not ionized. These are polar covalent bonds. So the amino group is going to be hydrophilic. And do remember that we said that there's a pair of non-bonding electrons right here with negative charges. If we have any protons, hydrogen ions in solutions, they can bind to those 2 electrons in a reversible ionization. We then have an ionized amino group, so it'd be NH3 plus because we have one more proton than electron. So we would have a decrease in hydrogen ion concentration, thus amino groups can act as bases. And we have a really cool word for substances which can act as an acid or a base. We call them amphoteric. So a free amino acid is amphoteric. It can act as an acid or a base. And then we'll be talking about the 20 to 21 unique amino acids found in proteins. What makes each of those amino acids different is this last group that's attached to our central carbon. And this is the unique R side chain or side group. And we'll see when we talk about amino acids and proteins that there are 20 to 21 different R side chains or side groups. Good. That's a great lecture exam question we just covered. So again, just our introduction to organic compounds. So we can take small organic molecules and combine them into larger, what we call macromolecules. Macro means big molecules. Often macromolecules are polymers. Polymers are chains, and these polymers consist of many small repeating molecules or subunits. And those smaller molecules or smaller subunits are called monomers. So mono means one, mer means part. Okay. So many of our big macromolecules then are polymers made of monomers. So since so many of our organic molecules are polymers, okay, poly meaning many, mer meaning parts, let's take a look at just some generic polymers. So in some polymers, for example, starch or cellulose, the monomers that are linked together to make the polymer can be the same. So for example, in cellulose, which is a polymer of glucose, glucose is linked together over and over and over again. But in other polymers, we can have a variety of different monomers linked together. So this is just our cartoon down here. And that's what we're going to see with proteins. Proteins are polymers of amino acids. And remember, there's 20 to 21 different amino acids. So the next question we want to ask, well, how do we take a monomer and link them together, right, to form these strong covalent bonds here? And as you might remember from chemistry, this is going to be done in a cool process called dehydration, synthesis, or a condensation reaction, which I think we have an example on the next slide. Good. So you guys, this is just a generic dehydration, synthesis, or condensation reaction. So over here are our reactants, right? And the arrow pointing to the right, this represents electron rearrangement. And then these would be our end products. Right, here's our end products. So reactants, electron rearrangement, and then end products. So in a dehydration, synthesis, or condensation reaction, we're going to remove the elements of water. So we're going to grab a hydroxyl group from this reactant, we'll grab a hydrogen from that reactant, and with electron rearrangement, those elements will be removed to form water. And then again, as that electron rearrangement, we're going to form a new covalent bond from this oxygen to the residue of that reactant. So here's the residue of our one reactant here, and here's that new covalent bond that was formed right there. Now the reason I put a double-headed arrow on this is that often condensation reactions or dehydration synthesis, they're reversible, meaning we could take this little guy, we could call this a dimer, we could reintroduce the elements of water with electron rearrangement, and we could hydrolyze that covalent bond, releasing the two original reactants here. So that's hydrolysis, and we'll be talking a lot about hydrolysis later. So the four big groups of organic macromolecules that we'll be discussing are the proteins, made of chains of amino acids, so polymers of amino acids, the nucleic acids, DNA and RNA, and their polymers of nucleotides, carbohydrates, and some of the carbohydrates, what we call the polysaccharides, are polymers of monomers made of monosaccharides. And then when we start talking about the lipids, we'll see lipids are kind of the exception. Lipids aren't polymers, but instead what makes lipids really cool is they're really hydrophobic. And the reason we're going to focus on these four groups is these are the building blocks of cells, and we'll also see that at least some of these macromolecules are the building blocks of viruses and prions. So I'm going to advance to the next slide, but we'll close this movie right here, trying to do these in little chunks. Okay, so the lecture unit 2, part 3, then we'll start with a discussion of proteins, and proteins are so much fun. They're so cool. Okay, so let me see if I can finish this one, and then we'll start with proteins on the next movie.